Welcome back, everyone, or welcome if this is your first time. This is week 2 of our HERC's Econometrics series. Last week, we had Todd Wagner talking a little bit about some of the problems that could arise in econometrics research. And this week, we have Laura Graham, who is a very experienced VA researcher, as well as at Stanford, talking about research design. And she is an epidemiologist by training. So even though this is an econometrics series, we've been promised a bit of epi-spin. And I will be handling the Q&A. So if you have those, please put them in there, and I'll either clear them up or get Laura to answer those for us. Go ahead and take it away, Laura.
Yay. Thank you so much, Liam. As Liam said, I am an epidemiologist by training, so we've updated this slide deck a little bit from prior years. So I do hope that you enjoy the updates. Definitely let us know if there's anything else we can add to it. But this year's edition is going to have more of an epi-spin on it. So, first, an overview. We'll talk a little bit about what research design actually means, the pros and cons of commonly used study design, and then we'll finish up with some discussion of measurement error and bias consideration. And in keeping with the seminar series, our focus is going to be on human subject research and quantitative research designs, which are things that you're most likely to see in HERC research and also in the VHA data. So hopefully this will be useful. Probably a little bit of an overview. Some of it is things that you've already heard in a review, but hopefully it does get you thinking about some of your research design questions. So we'll start out. I think Maria is going to help me a little bit with this. We've got a couple of poll questions just to find out kind of who you are, what you do, and how we can help as far as this research series.
Okay, the poll is open. What is your background? And check all that apply. We have clinical, biostatistics, epidemiology, economics, data science, other mathematics or science background, other non-mathematics or non-science background. And the results are coming in slowly. And it looks like I'm able to see the results. Hold on. We weren't able to see the results earlier. I'm going to give you just a few more seconds because they're still coming in rapidly. Okay, I'm going to go ahead and close that poll. And the results we have, 33% say a clinical, 19% are biostatistics, 21% say epidemiology, 23% say economics, 19% checked off data science, 19% checked off other mathematics, and 13% other non-mathematics. And I'm going to head and here we go. And back to you.
Awesome. So it's great to hear. We've got a nice mix going on. Hopefully, I won't bore the epidemiologists and economists too much. And hopefully, this will be helpful for some of the clinicians and other data scientists out there. The next question that I wanted to ask, just to get a feel for experience, was just how many years you've been working in research.
Okay, that poll is open. We have less than 2 years. We have 2 to 5 years, 5 to 10 years, and greater than 10 years. And the responses are coming in quick. And we'll just give it a few more seconds, and then I'll go ahead and close the poll. Okay, I'm going to go ahead and close that poll. And the responses are, we have 16% that say less than 2 years, 26% is 2 to 5 years, 19% 5 to 10 years, and 31% is greater than 10 years. And back to you.
Yay. Nice distributions here of experience and background. So I think that based on your characteristics, I hope this will be helpful. I think it will be. I kind of drafted it towards that mid-range of experience. So we'll talk a little bit first about research design overview. And what is it? Well, it's actually pretty simple. It's very straightforward framework or strategy or your plan to conduct research. We're all familiar with the scientific method. You have a question, you do a literature review, you have a hypothesis, and then you design your study. That's the research design part of it. Another good way to think of it is basically anything that you would include in a study method when you're writing for a grant or you're writing up a publication. And with that in mind, I wanted to drop a very quick reference here, and there's a couple of other resources at the end of the slide deck, but I think this is so helpful for thinking about research design, not only in econometrics, but also in other types of research out there. The Equator Network. And basically what this is set up for is it kind of collects a lot of the guidelines for publishing research. And these guidelines actually have great guidelines for a methods section in all of them. I highly recommend you check this out. I tend to use the Strove guidelines a lot, which are more epidemiology focused. But I ended up using this to think more so about the econometric side of research and what we're doing at HERC. And they actually have a guideline there called the Consolidated Health Economic Evaluation Reporting Standards. So these are the things they list that should be included in econometric method sections. Of course, this is for papers. So there's much more to consider in the research design phase, but this does give you a really nice outline of how you should be thinking about econometric research design and health services research design as well. And when you start breaking these pieces down and try to kind of organize them into groups, you'll see that for this one, as well as for a lot of the other research study designs, they tend to have three common core components. The first being the study design, the second being the way that you're handling the data, and the third being the analyses that you did. Since the majority of this seminar series is going to focus on different types of analyses and ways to think about them for your research designs, I'm going to give you a nice basic overview of study design from an epidemiologist, and then we'll talk a little bit about some of these data considerations, measurement error, bias, endogeneity, things that may come up while you're thinking about a research design. So, first and foremost, introduction. This is a basic breakdown of the type of research designs that you're going to run into. Of course, we're only going to talk about the quantitative research designs, but it is important to know the difference between quantitative and qualitative research, because sometimes the question you have may not fall into that quantitative bucket. So quantitative research tends to be, it's more formal, definitely objective, systematic processes. Whereas with qualitative, one of the main differences is that it's more subjective, still very systematic, and often more descriptions and applying meaning to experiences. So one of the other main differences is with quantitative analyses, you may be thinking about testing relationships or examining causal associations. Those are quantitative questions. Whereas if you're thinking more so about discovering a framework or exploring some kind of particular phenomenon that you've noticed, that's more qualitative in nature. And then lastly, what I use to really kind of drill down to what a quantitative or qualitative study is are you testing a theory or are you developing that theory? Of course, you can use these both in a mixed method study design, but for the purposes of this talk, we are going to drill down into that quantitative research design. And when you break that up, you'll know that there's observational and experimental. Of course, Todd talked a little bit about the randomized controls, which fit under experimental design, but most of what we end up doing tends to fall into that observational research category. You'll know from last week's talk, experimental or interventional designs tend to be the gold standard. And that's because they have higher quality evidence and better study validity. In these experimental studies, the investigator manipulates the conditions or assigns the treatment groups, exposed, non-exposed. One of the things to keep in mind about the experimental studies is that really they're only ethically permissible when adherence to the study protocol does not conflict with the subject's best interest. So an example would be, it's unethical to force someone to smoke and force someone else not to smoke. That would be an experimental study design. So observational study design came about due to these ethical and cost restrictions of experimental studies. In observational research, the investigator does not control the exposure. So subjects, excuse me, are self-selecting into their treatment group. So now we'll talk a bit more about the different types of observational research design. As I mentioned, unlike the experimental design, the investigator does not assign exposure status. Subjects are self-selecting into these groups. And because it relies heavily on an understanding of selection of subjects into the treatment groups, this is actually the source of a lot of our research design concerns, how these subjects are going into the treatment groups and what is the background on that. It's also important to keep in mind that while observational research is often less resource intensive, can be quicker, cost less money, also more available data that's easily accessible, they are still less valid than experimental design. However, they could actually be more effective for rare outcomes. When we think about observational research, there's two different types of studies. Most of the research that we do tends to fall into the analytical observational research. However, it's also important to keep in mind that there are descriptive studies. Cohort and case control studies are always analytic. Descriptive studies, an ecologic study or a cross-sectional study, could actually be analytic or descriptive, and a case series, always descriptive. So what's the difference between these? Well, as I'm sure we all have figured out at this point, if you're testing a hypothesis, it's an analytic study. Those are the case controls and cohort studies that we do. They quantify the direction and magnitude of association. Whereas a descriptive study is more generating the hypothesis. It's describing a pattern, place, time, or person in a population of interest. And the main thing to think about here is the descriptive studies do not have a comparison group. So if you are struggling with trying to figure out a study design, stop and think, you actually have a comparison group, because I cannot tell you how many times I've seen descriptive studies that don't have a comparison group described as an analytic, when really it's an ecologic study design or it's possibly a cross-sectional design, which are still beneficial, but not analytic study types. So within analytic observational study types, we're going to talk a little bit about the main two study types. We'll describe what they are, and then also go through some of the strengths and weaknesses of each. The first one we're going to talk about is going to be the cohort study. The definition of a cohort study, of course, the definition of a cohort is a well-defined group of subjects that are followed over time for an outcome of interest. The key here being followed over time. With a cohort study, research subjects are first identified by their exposure status and then followed for disease. Now, this can sometimes get a little tricky because there are two types of cohort studies. There's a prospective and a retrospective. Prospective, pretty straightforward. It's how you would think of a cohort study. You assess exposure and then follow patients to disease. However, with a retrospective study, you assess exposure after some people have already developed the disease. And so oftentimes, especially when we're dealing with some of the existing VA data sources, we may actually be using a retrospective cohort study design. It all depends on if you're identifying your sample by exposure status, or, as we'll see later with case control studies, by disease status. So some of the strengths of cohort studies, because by far, I think that cohort studies might, of course, I'm probably biased, but I do actually think that they have some of the stronger evidence in the VA, and I think it's actually far more useful with VA data. One of the strengths that you're going to see with them is that they are very good for establishing a temporal association between exposure and disease. The reason being, you're identifying exposure and then following to the disease. So you know that exposure occurred before disease. Also, because you're incorporating a time element in the study, you can measure incidence of disease. And because you're identifying subjects based off of exposure status, they are actually really good for rare exposures with common diseases and can look at multiple outcomes. So think in terms of having, we'll go with the most recent example of COVID, where it could be rare exposure to have coronavirus infection. So it'd be really hard to go through and just take a full cohort and look for those. However, if it's a tiny group, you identify a small group of coronavirus patients, match to a comparison group, and then look at diseases there, it's a much better study design. It's also important to think of these because prospective studies allow better control over sampling and better quality assessments over time. And this is actually really important in the VA data. If you're using existing data and some of your data elements are incomplete, inaccurate, or maybe measured in ways that are not ideal for answering your research question, a prospective cohort study design can help you incorporate better ways of addressing those data elements. So for example, if you want to look at, and you'll see this pop up again later, blood pressure measurements. If we just pull it from existing records, there's really no standard protocol for that. So if you just pull blood pressure measurements from the VA, you're going to get blood pressures that were taken from patients that have been sitting down for 5 minutes, taken with different types of cuffs, taken at different times of day. And so you're going to see a lot of error in that. However, if that's an important aspect of your study, you might want to design a prospective cohort study that has a standard protocol for blood pressure measurement and use that instead. So with that said, there's still some weaknesses to cohort studies that you need to keep in mind. So one issue is, and this is for all retrospective studies, the issue of recall bias. Another that you can run into, which cohort studies are, this is a big problem for them and they should always be considered, is loss to follow-up. This is especially important for prospective studies, but does also need to be considered for retrospective studies as well. And as you might have suspected, prospective cohort studies can definitely be more resource intensive. They tend to require a larger sample size, takes more time, and a longer follow-up. And because you're following patients to disease, they're really not that good for rare diseases or rare outcomes. If you're following a cohort of 50 patients looking for the outcome of looking for some rare infection, you may never actually see it. So with those in mind, the alternate to that cohort study is a case control study. So this is going to address some of the problems that you might run into with a cohort study that might be issues with your research question. We all know case control studies, research are identified by their disease status. So by default, a case control study is always retrospective. One of the key considerations, and I cannot stress this enough for case control studies, is how you select cases and controls. So you'll see this come up a lot, especially with matching and propensity scores, covariate adjustment. The goal that you are looking for is that your cases should be representative of all diseased subjects in the community. So not just cases that were hospitalized with disease or something along those lines. They should be representative of all disease subjects. And along that same line, your controls should be similar to the cases in all respects other than the disease in question. So they should be representative of all persons without disease in the population from which the cases are selected. And that's the clear epidemiology definition of it. The way that I like to think of this is that your controls should come from the same population as the cases. They should have the potential to become a case, but just didn't. And that's how you identify an appropriate comparison group in case control studies. Now, some of the strengths for a case control study are they're good for rare outcomes, whereas cohort studies, you're identifying by an exposure. They're good for rare exposures. With a case control study, you're identifying patients who have had an adverse, let's say an MI, and you can definitely go for more rare outcomes, identify them by that, and find a good comparison a lot easier than designing that study as a cohort study. They can also be less resource intensive, and this is really just because they're always retrospective by design. And also the opposite of a cohort study here, where cohorts are exposure and you can examine multiple outcomes. With a case control study, you're identifying your population by disease, but you have the potential to examine multiple exposures. And so case control studies are often useful for generating hypotheses about risk factors of an outcome variable. Now, some of the weaknesses that you're going to run into are going to be, they are definitely more prone to bias. And this has to do with how you're setting up your cases and controls. They also, because you do not have an estimate of time, they do not estimate incidence. And also, tying back to one of the strengths of a cohort study, one of the weaknesses of a case control study is they examine only one outcome. So that sums up probably the two main study designs that you're familiar with, the analytic observational study design, the cohort study design and the case control study design. So next, I'm going to spend just a little bit of time talking about some study designs that could be analytic, but also could be descriptive. The first one we're going to talk about here is a cross-sectional study design, which oftentimes trips some people up a little bit. So in a cross-sectional study design, both the exposure and outcome are assessed at the same point in time or over a short period of time. And what that means is if you are administering a survey and it takes you 2 months to administer your survey, that's not a follow-up study. It's still a cross-sectional study design. The reason being is that at the time that that participant fills out that survey and asks, do you smoke? Yes, no. Have you ever had lung cancer? Yes, no. The exposure and outcome is assessed at the same point in time for the individual. One of the strengths of cross-sectional study design is that they do provide a very nice point-in-time prevalence estimate. And this is actually, especially in today's environment where we have coronavirus, we can do a pretty quick, up-to-date point-in-time prevalence of how many people have the infection at a certain period in time. They also require less time to complete because it's all assessed at one point. So think along the lines of surveys, questionnaires, and interviews. And because you are assessing everything at one point in time, it avoids the problem of loss to follow-up that you may see in a cohort study design. Oftentimes, you'll see cross-sectional studies used at the beginning of a cohort or a clinical trial to attain baseline characteristics. We'll talk a little bit more about this blending of study designs as a hybrid study design in a couple of slides. Now, some of the weaknesses of cross-sectional studies is, and this is one of the main weaknesses that I want to stress as an epidemiologist, is they do not estimate incidence. They have no time component. And in addition, they provide far less evidence of causal relationship. And the reason being is that with a cross-sectional study design, temporality cannot be confirmed. You've assessed the exposure and the disease at the same point in time. So back to thinking about our smoking and lung cancer question. You can ask, do you smoke? Yes. Have you ever had lung cancer? Yes. We have no way of knowing if the smoking occurred before or after lung cancer. One of our other potentially analytic, sometimes also descriptive study designs is an ecological study. This is not too terribly different, the considerations that you need to keep in mind from a cross-sectional study, but the one thing that I do want you to pull out of this is that an ecological study design, I've been seeing more and more of these pop up. So this is where the unit of analysis is a group, not an individual. So this would be the unit of analysis is a neighborhood, the census tract, a state, or even comparing countries. So the result is aggregated measures that are either reported, which is a descriptive study design, or compared, which is an analytic study design. So ecologic studies, where they really shine is for rare diseases, or if you're studying some sort of large-scale public health intervention. So thinking in terms of the COVID vaccinations and looking at effectiveness of different strategies for implementing vaccinations. That would be a really good ecological study. However, and I cannot stress this enough, with ecological studies, you should always consider the potential for ecologic fallacy. I think it always exists. There's always some effectiveness in all ecological studies. And ecologic fallacy is when the relationship observed at the group level does not represent the relationship at the individual level. And this can occur, a lot of it is due to the relationship differs based on grouping levels. So if you were to look at the relationship of census tracts versus states, you might see a very different effect of exposure and outcome. And then last, the descriptive study design of case theories, which most people tend to look over, but I think it can be useful in health services research because we are starting to get into this more modern research environment of describing new disease processes and identifying and describing these rare manifestations or identifying some kind of emerging health condition. So it is important to keep this in mind. My best example here being that of AIDS. So the first 1,000 patients with AIDS was actually done as a case series. And within that, 73% were homosexual or bisexual males and 24% were injection drug users. So it does not require a formal comparison group to conclude that these groups are at a higher risk, that those are risk factors for AIDS. And that's a pretty quick study design to be able to get the information you need to start responding to some emerging health condition or new disease that's coming. So one of the strengths, like I said, cost-effective method, quick, to describe rare manifestations, new emerging diseases, but you do have to keep in mind this is one of the weakest study designs. It is purely descriptive. By default, it does not have a comparison group. And it's also the weakest form of evidence. Another thing, and this is something that I tend to think more about as an epidemiologist, is case theories could potentially give you some misleading or possibly suggest a plausible causal relationship when none really exist. And how that might happen is based on who you are sampling for your case series. If you happen to sample a group of patients and describe them in your case series that are very different from the overall population, that can lead to some misleading results. So do keep that in mind if you are doing a case series. And that concludes our description of the study designs. I wanted to include just a few slides here, thinking exercises to try and highlight some of the differences between the study designs. So this is a guess the study design question. And let's say we want to know if aspirin is associated with postoperative bleeding. Now we could also state that we want to know if postoperative bleeding is associated with prior aspirin use. So it really doesn't matter which way those occur in that statement. What matters for study design is how we did it. So we asked patients on the day of surgery if they took an aspirin that morning or the day before. And then we queried medical records for postoperative bleeding events in those patients. So if you think about that, the key here is we're identifying our group by exposure to aspirin. So this is a cohort study, and it's actually a prospective cohort study because really we didn't know at the time that we identified these subjects and assessed exposure if they were going to have an event. Now, another one to think about is this. So let's say we want to know if aspirin reduces the risk of becoming infected with SARS or coronavirus 2. So this is a relatively new disease. It's rare. In order to assess that, we decide, you know what, let's just send out a survey. We're going to ask about if you're using aspirin and also ask about SARS-CoV-2 infection. And this one, because we're assessing them both at the same time, it's a little easier, right? We can get a lot of individuals with one survey, but still, this is a cross-sectional study design. So we really can't make any assumptions about that temporality of if aspirin use really does lead to coronavirus infection, or the other way to think about it is, you know what, you get coronavirus infection, you might be more likely to take aspirin. So cross-sectional study design, some of the weaknesses there. And then the last two slides here, I really wish you could really devote an entire discussion to some of these hybrid study designs. It's really important to know that they exist. Just because your research question seems quantitative does not mean that you have to stick with a quantitative research design. Your entire study could actually combine a couple of different research designs. So an example, sticking within a quantitative research design, you may have a nested case control study within a cohort study. Or if we branch out even further, you can go into the mixed methods realm, which is super interesting and combines the benefits of both a qualitative research design and a quantitative research design. So again, this topic alone could be, there's classes, whole semester classes taught on these. So it's very broad, but I wanted to make sure to introduce it. And the time that you would consider it is if you have some issues of a single study design. So these are just 3 little examples of when that might pop up. One of the ones that we see most often, and this actually results in a mixed method study, is if your underlying hypothesis, when you do your literature search and you come up with your hypothesis, if it's really not well supported, then you might want to consider using a qualitative design to support and guide the findings of a subsequent quantitative study. This would be of course, mixed methods design. Another one, and this is something that pops up a lot as well, is if you don't have detailed disease information. So going back to the aspirin and MI question, if you think about using a retrospective cohort study with VA data, you think about how aspirin is defined, it's not really defined that well on if it was held or resumed, how it was managed around the time of surgery. So what you might consider doing is setting up a cohort study to identify whether it's associated, and then nesting a case control study in there where you actually pull in people who have had an MI and then match those in some way to a comparison group and look back and do a chart review. So that would be a nested case control study within a cohort. Another one is also if you have concern about case and control selection. So going back to that slide, we talked about case selection, how they need to be representative of population where controls are coming from. They need to be similar. If you're concerned about that, then you could possibly nest a case control design within a cohort to ensure that those cases and controls actually do come from the same population. So that concludes the first component of research design. I talked a little bit about the study design elements, and I hope that was useful. Now we're going to go into a little bit more.
Sorry, do you want to pause for questions, or do you want to wait until the very end?
Let's see, it's 11:34. Let's wait until the very end. This is maybe like another 10 minutes or so. So
you get it.
Hopefully this will help answer some of the questions that popped up in study designs. We'll see. How do we define error? So error is very straightforward, the difference between the observed result and the truth. And the goal of a good research design is to minimize this error. Now we think of error in 2 different ways. We think of error as random error, and we think of error as systematic error in study designs. So what's the difference here? Well, random error is oftentimes we use the words precision or reliability to describe random error. This is the degree to which our methods produce consistent results. So the key there being consistent results over time. I'd like to think of this a little bit like an archer trying to hit a target. So as long as those shots that they are taking are closely nested together, they have very little random error. An example, one that we talked about a little bit earlier too, is blood pressure measurements when there's not a standardized protocol. One thing to keep in mind is that random error is a problem of all research designs, so you should always consider it. Another issue that you can run into with research design, the other type of error, is systematic error. And systematic error is typically referred to as accuracy or validity. So you think back to that comment about the archer and aiming at a bullseye. Accuracy and validity is the ability to actually hit the bullseye. It's the closeness of a measured value to the truth. Or, in other words, the degree to which your study actually measures what it is supposed to measure. When we think about systematic error, the first one that pops into my head, at least as an epidemiologist, is bias. So bias is a systematic error in the design, conduct, or analysis of a study that results in a mistaken estimate of the exposure's effect on the risk of disease. We're going to talk a little bit more, drilling down into this idea of systematic error, which is a big issue for research design. We'll talk about selection bias, information bias, confounding, and then also endogeneity. The selection bias, and this is something that we talked about with study designs, you've got to think about where your population is coming from. This is an issue with your method of participant selection, something that's going to distort the exposure-outcome relationship from that present in the target population. So this is when what you see is not actually what's occurring in the target population. When would that happen? Well, you can have a non-response bias. That's when surveying by phone may systematically exclude patients without phones. Or you could end up with the cohort study lost follow-up bias, like we talked about. Patients without the exposure may actually be more likely not to complete the study. And then one that pops up a lot that you should definitely consider when thinking about your research design is confounding by indication, a form of selection bias, where healthier patients may be more likely to get a certain risky treatment than non-healthy patients. And then lastly, something else to think about, which doesn't really come up so much in observational studies. It's more of an experimental study design issue, but definitely still something to consider is volunteer bias, where patients that are affected by the disease may actually be more likely and more motivated to participate. Another form of systematic error is information bias. So this is also something that I put a lot of thought into, especially dealing with existing VA data. So information bias occurs when information is collected differently between two groups. So you get misclassification. And this, of course, because it's a bias, the systematic error, leads to an error in the conclusion of the association or the error in the results of the study. So information bias can be differential, which occurs when the level of misclassification differs between two groups. So let's say the level of how you assess glucose differs between those with diabetes and those without diabetes. Or it could be non-differential, where the level of misclassification doesn't really differ between two groups. So another form of bias is confounding. So confounding occurs when the observed result between exposure and disease differs from the truth because of the influence of a third variable is key here. In contrast, effect modification, which these terms are often used synonymously and they are not, effect modification is when the effect of the exposure is different among subgroups. So it's not a distortion of effect due to systematic error. It's simply a difference among subgroups. And to visually represent what confounding is, we usually look at figures like this. So confounding in epidemiology, you need to have three components. First, your confounding variable needs to be associated with both the exposure and the outcome. So you'll notice that these errors right here associated with both exposure and outcome. One of the other ones that's oftentimes missed is that it needs to be distributed unequally among those comparison groups. If it's distributed equally among those comparison groups, it doesn't matter. But if it's unequal distribution, that's a component of a confounder. And then lastly, it is not in the causal pathway. And what I mean by that is that these errors are pointing to exposure and to outcome, and that this confounding variable does not come from the exposure. The confounding and endogeneity. Last week, you heard Todd talk a little bit about endogeneity. And it's important to keep in mind that these are similar, but not the same. So endogeneity occurs when a variable in a multiple regression model is correlated with the error term. And this may be due, and I think this is partially why people tend to use them synonymously sometimes. Endogeneity can be due to an omitted variable, which is actually residual confounding at times. But endogeneity can also be due to measurement error of collected variables, and also simultaneity, which is when X causes Y, but Y also causes X. So you end up in this loop. So some of the design solutions that you can use to fix confounding and endogeneity got restricting the cohort, instrumental variables, matching comparison groups, which comes with its own set of cautions on that, and the whole talk could be devoted to matching, covariate adjustment, and then also randomization. And I'm putting this up here. I'm sure everybody on the call probably already knows these, but it's always useful to have it here just so that you can remember you can actually work to fix these in a research design, as long as you're thinking about them before starting the study. Garbage in, garbage out. And then the last three slides here, I wanted to introduce, and this is also another, like I said, this is very much an overview of research design topics, because I think directed acyclic graphs, really their own topic through and through, and could be their own presentation, but they're so useful for thinking about research design. And so I definitely encourage you to look them up. There's a couple of references and resources at the end of this presentation that you can use to get started looking them up. But they can help you really kind of conceptualize your research question. They are visual representations of causal assumptions of your research question. It's basically a conceptual framework that's unique to your research question. And they are great for illustrating some of these sources of bias that I just talked about. Confounding, endogeneity, looking for effect modification, and mediators. So some of the key factors of a directed acyclic graph, factors are connected with arrows. The arrows represent the direction of the causal relationship. That's key for the directed aspect. And acyclic, we talked a little bit about the simultaneity. Acyclic means that no directed path can form a closed loop. So a factor cannot cause itself in a directed acyclic graph. An example being this, our exposure being E, our outcome being D, C being our potential other variable. The figure that you see on the left is a DAG. The reason being, it's directed, it's acyclic because this right here ends in the disease. This one over here is not. And the reason being, you'll notice that this arrow right here has changed direction. So this is not a directed acyclic graph. And this one right here, we're actually looking at something that is a cyclic process. And for our final slide, and hopefully you'll find this useful, Like I said, they can be very, very helpful in identifying confounding and endogeneity during a study design. You can think about instrumental variables, how measured confounders, or possibly even residual confounding, things that you haven't measured, or as you'll notice here, mediators may affect your study question. And with that, we've got about 15 minutes to go, so perfect timing. The last little bit here being references and resources. As an epidemiologist, I always call on Modern Epidemiology by Ruckman. So that one's in here. And then a lot of the causal diagrams and the DAG information, highly recommend you check out Greenland's Causal Diagrams for Epidemiologic Research. And then lastly, I can't stress this enough, but do definitely check out the Equator Network, especially if you are about to submit a paper. It's just awesome for helping you organize your thoughts there. And with that, turn it back over to Liam. Ready for questions?
Yeah, we got a good amount of them to go through. So let's go back to case controls here. If you choose your the question is, if you, in the case control, if you choose your subjects by disease status, but then look at their treatments going forward, can they be seen as prospective?
That is a really interesting question. Yeah, technically you could. But then again, that also brings, I would question, what is your outcome? Because at that point, you may be choosing patients by your disease status. But if you're thinking about a change in outcome, then really by looking at the change in exposure or treatments going forward, you've actually created a cohort study nested within some kind of case control study.
Okay, great. Does case series result in a treatment tree or diagram? This is our next question.
I'm not familiar. What is TREE-er?
Treatment tree or diagram.
Oh, you could, but not necessarily. The main case series, it's just basically a descriptive report.
Got it. And do you think the next question, do you think you could describe the difference and maybe give some examples of a difference between case control and retrospective?
Yes, I know. That is always, so even I to this day will struggle with that. And I think that can also lead to some debate too, because it's very small differences. The way that I like to think about it is that most of the time what you're looking for is going to be a retrospective cohort, especially in the VA. Think about where the main emphasis of your research question lies. So I'm trying to think of some examples. Like one of the ones that I've done prior has been looking at the effect of different types of cardiovascular stents on surgical outcomes. And what we ended up doing was we just identified everybody who had surgery and then looked for stent status and surgical outcomes in that. And that is definitely a cohort study. They had the exposure before they had surgery. We ensured that. And then we also really emphasized our exposure of cardiovascular stent. Another way to think about it is how you think about presenting your results. But one of my pet peeves is always that odds ratios are really more suited for a case control study. And if you're focusing more on this element of time, then you're definitely probably looking at a retrospective cohort study. So hopefully that helps. But yeah, you get into some really interesting nuances when you start thinking about that, for sure.
Yeah, so I always had the impression it's like just depending if you're looking at trying to figure out the factors associated with this exposure or the factors associated with getting the disease. How would you think about that?
So if you're thinking about factors associated with exposure, then that's really more so in a cohort study realm, right? Because you're thinking about probably things that are preceding the exposure. In that case, your exposure is actually really just a disease. or it's your outcome of interest. It's your dependent variable. Whereas if you're thinking about, I mean, the straightforward exposure factors that influence disease, one way you might also break that down is if you're interested in looking at multiple factors that may influence that disease, then you might actually be thinking about a case control study. Does that kind of help?
Yeah, just trying to get, more into the differences between the two.
Yeah, getting back to that whole concept of cohorts are much better for multiple outcomes, whereas the case controls are much better for multiple exposures.
Our next question, could you give an example or maybe a few of information bias? That's something you mentioned a little while ago.
Information bias, yes, gladly. That is definitely something that pops up a lot. Let me pull up that slide just so we've got it going. Okay. So thinking about information bias, and the one that I think is most important is differential misclassification, where the one that I talked about, or I can't remember if I mentioned it, but possibly measuring levels of glucose. And if glucose and you're looking at the risk of death, well, think about it in terms of measurements of glucose in somebody with diabetes is far different from measuring glucose in somebody without diabetes. And so it's this differential misclassification by diabetes that could affect your association with the disease, in this case being mortality.
Okay, great. So this is not related to that. The next question is, if the survey asks for childhood trauma before 18 years old and the current health outcome, would that be cross-sectional or retrospective cohort?
That is a hard question. It depends on how strongly you believe that you can infer temporality out of that. I would lean towards cross-sectional just because I think it's very hard for you to confirm as the investigator that the exposure and outcome, and there's also issues of recall bias that you can see with respondents. But if you have a way of confirming that, yes, this exposure happened after childhood, such as an incident, like maybe you go and check the medical chart, you're able to determine when the disease actually occurred, then yeah, you could potentially call that a retrospective cohort study.
Okay, and the next thing is maybe talk a bit more about mediation. About how mediation is related to exposure and disease, and how mediation is different from confounding.
That is a whole talk, and I am horrible with mediators. So whoever had that question, I would be more than glad to discuss with you via email, but it would be a mess for me to talk about mediators and confounders.
Yeah, I guess I'll just mention that we do have a separate talk on instrumental variables, which is really the way that the language that economists and econometricians would use for mediators, for the most part, mediators and confounding. So that's kind of the solution to that problem. And I think that talk is in 2 or 3 weeks. I can't quite remember, but I would recommend you attend that one. Okay, so our next question is, a retrospective cohort study does not have a control group. A case control group has a case control has a control group, even though it is retrospective. Would you say that's right?
No. So this kind of gets back to the discussion of what's the definition of a control group. And this can always be I see this it's the terminology always trips people up. So the goal of the retrospective cohort study, both of them have a comparison group. It's just in the case control study, that control group is what we call the comparison group. And within a retrospective cohort study, the control group is actually the non-exposed group. And so you don't tend to hear non-exposed. You might hear the comparison group used for a retrospective cohort study, but ultimately the control group in the retrospective cohort study is still a comparison group. It's just non-exposed as opposed to the control group in a case control, which is non-diseased.
Okay, great. So yeah, we worked through those questions. I think we have like 5 minutes. I think maybe a really interesting thing to talk about then would be how, as an epidemiologist, you've been thinking about the way that COVID research has gone and what kind of studies you think have been more effective than others as you've seen them all come rolling out.
Yeah, definitely. I think this has been an interesting time for epidemiologists because we really get to see how we use these different study designs. And I think in times when you're not dealing with like emerging and new diseases, we tend to rely heavily on these cohort and case control studies because we've got time and we've got data that we're able to work with. But one of the most interesting things that I think we've seen with the pandemic as it's progressed is very early on, we didn't have time to figure out exactly, we needed information and we needed it now. And so that's where you started seeing a lot of these like case series pop up. And I think that really does show the importance of a case series design. When you're dealing with something that you have limited time, you need to get the information out there because you need to start acting. Right now, I think what we're starting to see that's the more beneficial, of course, we've got lots of experimental design because we're trying to take the case series information and figure out how to act on it. And we don't have any really major ethical issues right now. So the experimental designs really, I think, are the highlight of all of this COVID research that's going on. However, moving on, I do think we're going to see a lot more of these prospective cohort studies, not just looking at how to prevent COVID, but also looking at kind of what influences disease severity and also long-term outcomes.
Okay, great. That's a really interesting answer. Thank you for that. And we don't have any further questions, but if you go back to the last slide, which are available not just here, but in an email, as Maria has reminded us, Laura's email is in there. You can, I'm sure, email her with questions. Also plug that, HERC has a help desk. So if you can also ask us about questions, especially about if you're a VA researcher, but we're also happy to talk about other things. And I'll also plug that we have another further, I think, 9 more of these with various topics, a lot of them having to do with what you do when you encounter problems with your study in econometrics. And I think that's it. So thank you very much, Laura, for this great talk.
Thank you.